All right, so this talk is about single trait crosses. All right, so how to do a punnett square that crosses the genes for a single break. Punnett square is 2 T's. Sorry about that. There we go. Okay. All right. So the first thing you need to know is Mendel laws that support this, because he's the one that came up with the laws of inheritance that allow us to even do it. And the amazing thing about Gregor Mendel, who was working in the 1800s, about the time the United States was having its American Civil War, he was figuring out these inheritance patterns before we even knew what chromosomes looked like, or that DNA was inside chromosomes, or that meiosis was the thing that happened. Nobody even knew any of that yet when he discovered these patterns. So the first thing that he discovered was something called the law of dominance, okay, which basically says every characteristic is controlled by 2 alleles, and one tends to dominate over the other. All right, so what does that mean? You might have in his pea plants, right? In pea plants, big T was the allele for tallness, and little t was the allele for shortness. So what would happen? Big T would give what kind of a plant, tall or short? It would give tall. Good. Big T, little t would give what kind of a plant? Tall again, as big T dominates over little t. And little t would give short. Very good. Okay. Assuming that you guys are okay, you're seeing everything. Yes, and you're participating. Beautiful. Okay. All right. So the other words that go with this are the words genotype, which would be the letters, or actual genes. Example, little t would be a genotype. And phenotype, which is the physical description that's as a result. Example. Okay. The law of segregation is the next one. Law of segregation means that during gamete formation, and again, he didn't know that this was meiosis. He had no idea. But during the formation of sperm and egg cells, the alleles for each trait separate before then reappearing in offspring. Okay. Here, I want to actually draw a picture of what that means, which is why I was doing this. Okay. Okay. So, for example, if the parent generation, which is always pure, let's say you had a pure tall plant times a heterozygous tall plant. Remember the words homozygous and heterozygous. I'm kind of going over this quickly, but I want you to get to work, and I don't think you really need this. I have a feeling you're going to remember it so fast you're not going to need to go over this too much. But let's say you have these parents, and you know that a Punnett square is a way to predict the probability of parents' offspring being certain genotypes and certain phenotypes. Let's put the homozygous dominant parent here and the heterozygous parent over here, and this is segregation, that the alleles separate and go here across the sides of the Punnett square, right, singly. That's what segregation looks like. Segregation is the separation of alleles before fertilization, right? So, for example, these guys might be in the sperm created by this plant. Yes, plants make sperm. And these guys might be in the eggs created by this other parent plant. Yes, plants make eggs. And then when things are carried through, what's in each of these boxes is the potential offspring from these two parents. So now let me ask you this. The predicted, right? The predicted offspring. Each one of the genotypes in these boxes is a possible offspring from these two parents. So what is the percentage of these guys who are going to be big T, big T? %, very good. And what about the percentage of heterozygous offspring? Again, 50%, very good. Okay, what about the percentage of short offspring? 0, very good. None of these offspring can be short because all of them have big T's in them. What about the percentage of tall offspring? 100, exactly. Okay. Remembering how all this works? Yes? Good.